Welcome back to the deep dive. Our job here is to take the confusing, contradictory, and often overwhelming world of research and really distill it into the essential knowledge you need to feel genuinely informed.
In today's topic, it really does venture into a corner of science that sounds, well, less like medicine and more like science fiction.
It absolutely flips the script on conventional wisdom, doesn't it? I mean, about health, cleanliness, disease.
For centuries, our entire public health focus has been on eradication and hygiene. This deep dive asks, what if that's success? What if it's also the source of a massive modern health crisis?
Exactly. We are talking about Helminth therapy. The idea that parasitic worms, our supposed biological enemies, might actually be a crucial missing biological regulatory component.
A component we need to treat the chronic inflammatory diseases that have just exploded across developed nations.
We are trading ultrasanitation for symbiosis, and the implications are, well, they're staggering.
Our mission today is to dive deep into the evolutionary and molecular basis for this. We'll start with the famous, or maybe infamous, hygiene hypothesis, and the more specific, old friends mechanism.
And from there, trace the loss of these organisms historically, right?
Right. And then dissect exactly how these worms hack our immune system at a cellular level. Ultimately, we want to know why diseases like Crohn's and ulcerative colitis, and surprisingly, even conditions linked to stress and poor mental health, are becoming sort of the default condition of high-income urban living.
Okay, so let's start by establishing the gravity of the problem we are attempting to solve. We really need to understand the full weight of inflammatory bowel disease, or IBD.
Which covers both Crohn's disease and ulcerative colitis.
Right, and when we talk about IBD, we're talking about a chronic relapsing inflammation of the gastrointestinal tract whose origin is idiopathic, meaning, essentially, we don't know the cause.
We don't. But the symptoms are devastatingly real. IBD isn't just a digestive annoyance. It is a global health crisis that has reached staggering levels of prevalence, hitting up to 400 per 100,000 persons worldwide.
And while Crohn's can affect any part of the GI tract, ulcerative colitis, or UC, is typically confined to the colon and rectum.
And its symptoms are particularly brutal and life-altering for the patient.
Can you walk us through what those specific symptoms of UC look like? Because the numbers in the materials are, frankly, hard to grasp without context.
Absolutely. Think about what chronic uncontrollable inflammation does to the lining of the colon. Patients suffer from a progressive loosening of stools, often bloody, relentless diarrhea, and tenesmus.
What's tenesmus?
It's the constant painful feeling of needing to evacuate your bowels, even when they're completely empty.
Wow.
And for severe sufferers, this can mean a terrifying, severe urgency to go to the bathroom up to 15 or even 20 times a day.
20 times a day? That doesn't just impact your diet. That impacts every single decision you make.
Every single one. Where you work, whether you can leave the house, whether you can sustain a normal relationship, it completely dictates the structure of your life.
It's utterly life-limiting. And it often strikes people in the prime of their working lives, which is just tragic.
It is. And when we look at how we treat this condition currently, we immediately see the profound need for a new approach.
The current medical toolbox for IBD, it seems less like a cure and more like a series of increasingly difficult compromises.
It's a perfect way to describe it. Current therapy focuses entirely on achieving and maintaining remission, not curing the underlying immune problem.
So you start with what? Anti-inflammatories?
We start with aminosalicylates, which are anti-inflammatory drugs. If those fail, we move quickly to steroids. Steroids like prednisone are incredibly effective at knocking back acute inflammation, but they come with a brutal price tag.
Oh, the side effects are notorious.
They are. They can cause bone density loss, mood changes, insomnia, and they can only be used short-term. It's a rescue drug, not a maintenance drug.
And after the steroids fail, you're usually looking at heavy-duty immunosuppressants.
Exactly. We're talking about immune modifiers like azathioprine or methotrexate. These drugs suppress the entire immune system just to quiet the gut inflammation.
And the obvious downside there is systemic immune suppression, making you vulnerable to all sorts of infections.
Yes, and even worse, many patients become refractory, meaning unresponsive to these treatments over time. Their body just stops responding.
And the long-term prognosis for ulcerative colitis carries a truly terrifying risk, especially concerning cancer.
Yeah.
Can you put that risk into perspective for us?
If a patient has had UC for a prolonged period, typically 7 to 10 years, they face a dramatically increased risk of colonic epithelial dysplasia and carcinoma.
And that risk just keeps climbing over time.
It increases relentlessly, approximately 0.5% to 1% per year after that initial period. That means 10 years after diagnosis, the cumulative risk is significant, requiring constant surveillance.
That's why surgery becomes, tragically common.
Precisely. Despite all these pharmacological attempts and the associated risks, up to 25% of patients with ulcerative colitis will eventually require a colectomy.
The surgical removal of the colon.
Yes. And this treatment gap, the fact that our best medicines are compromises that fail a quarter of patients and carry serious long-term risks, that is the central justification for researchers looking at radical, unconventional options, including the reintroduction of parasitic worms.
So let's unpack the core mystery. We know IBD is associated with a hyperactive, deregulated pro-inflammatory response to our own gut bacteria.
The friendly commensals that should be ignored, yeah.
But why are modern immune systems failing to regulate this response? Why is this happening now?
Well, the current consensus is that IBD is a failure of immune tolerance, driven by a complex interplay of two factors, genetics and environment.
On the genetic side, what have we found?
We've identified around 163 genes that are crucial for IBD susceptibility. Many are involved in how the immune system processes and responds to bacteria.
So, some people are predisposed, but the genes only load the gun. The environment is what pulls the trigger.
That's a great analogy. The environmental triggers are so diverse. Diet, stress, heavy antibiotic use, the composition of our intestinal microbiota, they all conspire to disrupt that crucial immune balance.
And here's where the data often gets so counterintuitive, specifically with smoking. It seems to play two completely different roles depending on the disease.
It's a complete biological puzzle box. Research consistently shows that smoking increases the risk of developing Crohn's disease.
Okay, that makes sense.
But for reasons we still don't entirely understand, it seems to offer a protective effect against ulcerative colitis.
That's bizarre. A reversal?
It is. It suggests that the specific pathways leading to CD and UC, while overlapping, involve different molecular mechanisms. Perhaps it's the effects of nicotine or other smoke components on the gut lining, or the vasculature, or the microbiome. We don't know.
But regardless of the mechanism, it hammers home how exquisitely sensitive this gut immune system is to external inputs.
Absolutely.
Yeah.
And this perfect storm of genetic vulnerability and modern inputs brings us to the centerpiece of this discussion, the hygiene hypothesis.
A phrase coined by the epidemiologist David Strachan back in 1989. What was his core observation?
Strachan was looking at allergy rates, particularly hay fever, and he noticed something really striking. He hypothesized that the rising standard of living, coupled with smaller family sizes and increased hygiene, specifically the avoidance of common childhood infections, meant that the immune system was being deprived of essential training.
So the idea is that when the immune system doesn't have real enemies to fight early in life, it essentially gets bored and starts fighting itself.
Or it starts reacting violently to harmless things like pollen and peanuts.
Right.
Precisely. The hypothesis posits that exceedingly high cleaning standards and antibiotic overuse deprive the immune system of essential benign microbial exposure. This lack of early life actuation or practice results in a failure of regulatory mechanisms later in life, driving up immunological disorders like asthma, allergies, and IBD.
But the hygiene hypothesis is sometimes criticized for being a bit too simplistic. It makes it sound like dirt alone is the cure.
And that's why researchers evolved the concept into the old friends mechanism.
How does that provide the deeper evolutionary context?
The old friends mechanism provides the necessary evolutionary depth. It moves beyond simple dirt and focuses on specific, ancient, co-evolved organisms. For millions of years, mammals, including us, were guaranteed to encounter a specific array of microbial companions.
Chronic, non-lethal infections.
Exactly. Certain environmental organisms from mud and water, and critically, helminths.
And what you're saying is our immune system didn't just tolerate these organisms, it was fundamentally built to interact with them. They are like biological furniture.
That's the key distinction. Helminths, or parasitic worms, are long-term residents. They don't want to kill their host quickly. They want to live comfortably for years.
It's bad for business to kill the host.
A terrible evolutionary strategy. So to achieve this longevity, they evolved an incredibly sophisticated ability to chemically manipulate the host's immune system, forcing the development of complex immunosuppressive and regulatory circuits.
So when we remove these guaranteed old friends, we don't just lose the worms. We lose the essential inducers of that whole immunoregulatory network.
A network constructed over millennia.
Yes, and this loss is so recent in evolutionary terms. We went from being universally infected to being essentially sterile in barely a century.
The historical context is truly powerful proof. We have concrete data on how rapidly we erase these old friends. Consider hookworm.
Okay.
In U.S. schoolchildren in 1910, the prevalence was about 65%. 65%.
Wow.
By 1980, thanks to massive public health campaigns, sanitation infrastructure, and anti-Helminth drugs, that number was less than 2%.
We achieved a public health miracle, but we inadvertently created an immunological deficiency.
We threw out the regulator with the bathwater. This is why we also hear the terms lost friends theory and biome depletion theory. It emphasizes that the problem is the deprivation of necessary signals.
And the result of this deprivation is the massive epidemiological parallel we see today. The simultaneous steep increase in conditions like IBD, multiple sclerosis, type 1 diabetes, and allergies.
All these conditions correlate perfectly with economic development and urbanization dating back to the 19th century in Europe and accelerating globally ever since.
And if you need proof that the environment, not just genetics, is the major driver, you look at the migration data.
This is arguably the most powerful evidence we have.
So walk us through that migration risk. How does moving from a low-income country where you are likely exposed to these old friends change your disease risk when you move to a high-income urban environment?
It acts as a profound risk modifier. Studies globally show that moving from a high-exposure environment to a hyperhygienic urban one significantly increases the risk of developing these inflammatory diseases.
And it matters when you move, right?
It matters immensely. This is particularly true if the individual is exposed to the clean environment during the perinatal period or early childhood, that critical window of immune system training.
So someone who moves as an adult retains some of that original protection.
Often, yes.
Yeah.
Researchers found that first-generation immigrants often retain some degree of partial protection, presumably because their immune system was initially trained in their country of origin. But the disease prevalence increases in second-generation immigrants.
To the point where it can even exceed the native population.
Exactly. Often reaching or even exceeding the rate of the native population. We see this in studies on IBD risk in immigrants to Sweden and the increased prevalence of UC in South Asian immigrants to the UK. It tells us that those early life microbial exposures are the key to setting the long-term immune system's baseline of tolerance.
So if the old friends deficit results in a failure of immune system training, that failure shouldn't be limited to the physical location of the gut. It should be a systemic failure of regulation, affecting inflammation everywhere in the body.
That is the essential insight that takes this research beyond just gut health. If the background noise of systemic inflammation increases because the regulatory mechanisms the peacekeepers are missing, it affects the whole host.
Which explains the parallel rise not just in gut and autoimmune disorders, but also in certain chronic metabolic and psychiatric disorders in wealthy countries.
Precisely.
This brings us to a topic that is becoming increasingly clear: the tight link between chronic inflammation and psychiatric health, especially clinical depression.
The connection is undeniable. Inflammation is much more than just a localized protective mechanism. These inflammatory mediators, these signaling molecules, they actively modulate brain development, cognition, and mood.
And we now know that a large subset of depressed individuals aren't just chemically imbalanced.
Not at all. They have persistently raised levels of inflammatory markers in their bloodstream, specifically C-reactive protein, CRP, and interleukin-6, IL-6.
So some depression isn't caused by a serotonin deficit. It might be caused by an inflammation surplus.
Exactly. And the old friends deficit feeds right into this. The concept is that defective background immunoregulation exaggerates our physiological responses to stress.
So if your peacekeepers are absent, any psychosocial stressor, whether it's poverty, chronic life events, or perceived social threat, results in a disproportionately greater and more prolonged inflammatory surge.
That's right.
This sounds like a biological definition of reduced stress resilience. Our bodies are just overreacting to normal modern stressors.
They are. The immune system, lacking its natural negative feedback loop, can't terminate the stress-induced inflammation efficiently. The research on early life trauma proves this point powerfully.
Walk us through that specific data point on childhood maltreatment.
Okay, so studies show that adults who were exposed to early life psychosocial stressors, like childhood maltreatment or severe neglect, exhibit a profoundly exaggerated inflammatory response when they're later subjected to a standardized laboratory stressor.
What happens when they measure their blood?
They see much higher and more prolonged production of IL-6, for example, compared to control subjects. This points not just to trauma, but to a diminished fundamental ability to control inflammation.
Which is precisely the role the old friends were supposed to program into the system.
It's a tragedy layered on a tragedy. The loss of these evolutionary companions synergizes with modern psychosocial stress, exacerbating the inflammatory pathway that then drives psychiatric symptoms. It's the perfect failure of modern living.
And this susceptibility is locked in very early. We are talking about the perinatal window.
Events surrounding pregnancy and the first few weeks of life have massive long-term consequences for immune regulation and stress response.
So how does maternal inflammation or stress during pregnancy affect the developing fetus?
Well, maternal stress during pregnancy is associated with raised circulating pro-inflammatory markers in the mother, things like IL-6 and tumor necrosis factor, or TNF.
And those don't just stop at the placenta.
No, they don't. They can affect the development of the offspring's central nervous system and their hypothalamic pituitary adrenal axis, the HPA axis, which is the system that regulates stress hormones. This sets the stage for long-lasting immunoregulatory issues, making the child hyper-responsive to stress later in life.
And the microbial environment is also setting the course at the exact same point.
Critically so. Stress itself, whether it's maternal or environmental, modulates the gut microbiota, and the composition of the microbiota established during those first few weeks of life when the immune system is actively learning has persistent effects on the CNS and HPA axis.
And you can't go back and fix it later.
Not fully. What researchers have found in animal models is that if you miss those critical early microbial exposures, you cannot fully correct the resulting stress hyperreactivity simply by reconstituting the microbiota in adult animals. The window for programming the system closes.
It's fascinating how the epidemiological data circles back to this. The prevalence of depression, schizophrenia, even some spectrum disorders, they all tend to be higher in urban versus rural communities.
And the risk increases in immigrants who move during infancy or in second-generation immigrants.
It seems all roads lead to a common mechanism, a profound and system-wide failure of the immune system's negative feedback loop.
That's it.
Okay, let's drill down into the microscopic reality. If IBD is a gut civil war driven by a hyperactive immune system, and helminths are the missing peace treaty, we need to know exactly how they achieved that peace. We need the brass tacks of the hack.
Okay, so let's contrast the two states. In a healthy gut, the mucosal immune system has a dual mandate: control symbiotic bacteria and eradicate true pathogens.
But in IBD, the epithelial barrier breaks down and the immune system misidentifies harmless commensal bacteria as invaders.
Exactly. And that hostile response is driven by specific cells we call Th1 cells.
The shock troops of the immune system.
That's right. The chronic inflammation in IBD is aggressively driven by T helper 1 cells, Th1 cells. They're designed for acute, immediate, cell-mediated attack. They churn out aggressive cytokines.
What are those molecules of fire that they are producing?
They produce interferon gamma, IFNO, tumor necrosis factor alpha, TNFO, and interleukin-17A, IL-17A. These are the chemical signals that recruit more inflammatory cells and actively maintain that chronic state of inflammation. If you can't turn off the TNFO, you can't stop the gut fire.
Now enter the helminth. The worm is a long-term resident. It needs a calm, tolerant environment to thrive. How does it shift the immunological ecosystem away from this aggressive Th1 response?
The Helminth strategy is sophisticated and compulsory. It forces the immune system to shift entirely away from the Th1 fire and toward a T helper 2 response, Th2.
Okay, a Th2 response. What's that?
The H2 cells specialize in clearing parasites, but in the process, they produce different, more regulatory and healing cytokines, IL-4, IL-5, IL-10, and IL-13. And crucially, this shift massively expands the population of regulatory T cells, or TREGs.
The TREGs.
Yeah.
We've called them the police force of the immune system before. They are the ones who patrol the borders and tell the attack cells to stand down.
They are the ultimate controllers of immune homeostasis, especially in the intestine. Defects in these FOXP3 plus TREGs are directly implicated in autoimmune conditions and IBD. The helminth is a master at forcing the creation and expansion of these TREGs.
How does the worm achieve this induction? It sounds like it's flipping an internal switch.
It's flipping a switch, and it involves one key anti-inflammatory molecule, transforming growth factor beta, or TGF-β.
TGF-β.
Helminths stimulate the production of TGF-β in the intestinal mucosa, and high-level TGF signaling is absolutely essential for inducing CD4 plus T cells to convert into regulatory T cells that produce the soothing cytokine, IL-10.
But the body has a natural way of blocking this conversion when it doesn't want tolerance, right? It has an internal brake system.
Precisely. Under normal, uninfected, clean conditions, intestinal T cells express high levels of a protein called SMAD7.
SMAD7.
You can think of SMH7 as the host's built-in emergency brake on tolerance. It actively blocks the TGF signaling pathway. It says, no, we are not going to create regulatory cells right now.
And this is the core of the Helminth's hack.
This is the moment of genius. Helminth infection actively and consistently inhibits the expression of SMAD7 in gut T cells.
It takes the brake off.
It takes the brake, SMAD7, out of the equation, allowing the powerful TGF signal to proceed unimpeded. This results in the massive, sustained induction of anti-inflammatory TREGs, which then flood the gut and successfully suppress the colitis that was driven by the aggressive Th1 cells.
That is an incredibly specific and sophisticated piece of chemical engineering. The Helminth has evolved a mechanism to disable a key component of our immune regulation.
A component that has been in place for millions of years. It's forcing the body to prioritize tolerance over attack.
And this immune hack doesn't operate in isolation. You said the Helminths are also in constant, complex dialogue with the other old friends, the massive microbial community in our gut.
Yes, they are ecosystem engineers.
So how does the presence of a macroscopic parasite fundamentally change the microscopic world of the gut microbiota?
It's consistently shown that Helminth infection profoundly alters the gut microbiota composition, and generally, these alterations favor beneficial bacteria. This is a critical point. The therapeutic effect may not be just the molecules the worm secretes.
But the entire new microbial community the worm enables.
Exactly.
Give us some concrete examples of those microbial changes.
In animal models, infection with the Helminth Lignosoides polygyrus is shown to significantly increase the abundance of Lactobacilli and Lactococcus. Trichuris suis infection similarly promotes lactobacilli.
And we know lactobacilli species are associated with anti-inflammatory properties and mucosal healing.
We do, but it gets even more dramatic when you look at genetically susceptible hosts. In mice engineered to lack the NOD2 gene, a key genetic vulnerability in many Crohn's patients. Trichuris infection helps restore a crucial defense mechanism,
Which is?
the goblet cells, which produce protective mucus. This restoration prevents the dangerous overgrowth of harmful pro-inflammatory species like bacteroids, while favoring protective bacteria, specifically the Clostridiales.
So the Helminth acts as a kind of biological shepherd, shaping the environment to encourage the good bacteria and inhibit the bad, especially in people who are genetically vulnerable to this gut civil war.
Absolutely. And the corollary is frightening. Studies show that when genetically vulnerable individuals are dewormed, losing their helminths, they show a shift in their microbiota that promotes the conditions associated with Crohn's disease.
So how do these ecosystem engineers communicate these complex anti-inflammatory instructions? It must be through specialized chemical language.
It is. This language is carried by what researchers call immunomodulatory components of helminths, or ICHs.
ICHs.
Helminths secrete these molecules via excretory-secretory ES products. These products are a complex soup of molecules, including antimicrobial peptides that directly influence the microbiota, but most importantly, highly complex post-translational modifications.
What makes these modifications, the glycans and phosphocholine groups, so potent compared to just a standard protein?
Well, these are not simple peptides that you can easily synthesize. These are highly complex sugar groups, glycans, and lipid modifications like phosphocholine that are recognized as the active components in the interaction with the host immune system.
They're evolved to be the perfect key for our locks.
A perfect way to put it. They've evolved over millions of years to perfectly bind to and regulate host immune cell function. They specifically suppress macrophage activation, which is a major source of early inflammation, and facilitate the massive induction of those crucial TREGs. They are highly complex, perfectly tuned signals designed to communicate tolerance.
The scientific mechanism, the SMAD7 hack, the TREG expansion, the microbial engineering, it's incredibly compelling. But moving from this beautiful theory to clinical reality is often where things, they break down. What happens when researchers actually give people live worms?
The clinical trials have focused mainly on two species of parasitic nematodes, or roundworms. The first is Trichuris sus ova, or TSO, which is the pig whipworm egg.
Why the pig whipworm specifically? Why not a human parasite?
That's the elegant solution to the safety problem. T. suis can infect humans, but crucially, it's not biologically adapted to us. It only survives in the human digestive tract for a few weeks before being cleared naturally.
So you get the immune signal without a long-term infection.
Exactly. It minimizes the pathogenic risk. The second primary species is Necator americanus, or Na, the human hookworm, which is administered safely via dermal exposure, basically applied to the skin.
And those early open-label trials were extremely promising, weren't they? They painted a picture of dramatic success in IBD patients.
They generated huge excitement. For example, open-label studies using TSO in Crohn's patients reported significant clinical improvement in around 79% of participants. 79%.
And for ulcerative colitis.
A small controlled trial in UC patients showed a significant improvement in 43% of Helminth-exposed patients versus only 17% on placebo. It'll look like a miracle cure.
But then the large-scale, randomized controlled trials, the scientific gold standard came in, and the results were much more complicated.
This is where the translation challenge became apparent. Later, large RCTs, such as the major TRUST-I and TRUST-II trials for Crohn's disease using TSO, failed to show clear statistical significance over placebo.
Wait, if the biological mechanism is so strong, why did the large trials fail to separate TSO from the placebo? What happened?
There are a few likely reasons, but the primary statistical challenge was the strikingly high placebo response rate. In many IBD trials, patients in the control group report clinical remission rates as high as 40% to 50%. This is likely due to the intense scrutiny, the psychological support, and the lifestyle changes that accompany just being in a trial.
So when your placebo group is showing a 50% success rate, it becomes statistically almost impossible for the active agent to show a significant difference, even if it is biologically active.
It gets incredibly difficult.
But despite the mixed efficacy data, there seems to be one consistent, crucial finding.
Absolutely. The safety consensus is the cornerstone of this field's legitimacy. A systematic review of all 9 major clinical studies consistently concluded that Helminth therapy, using both TSO and Na, was safe and tolerable for IBD patients.
So the side effects were minor.
Very mild, usually transient flatulence, diarrhea, minor abdominal pain, or mild itching at the hookworm application site.
But you mentioned that using live organisms, even low-risk ones, does carry small but real drawbacks.
It does. We can't ignore the risk, however remote. One patient in a trial, for instance, developed a live, sexually mature T. suis infection, which, while manageable, is the exact outcome researchers want to avoid.
And hookworm has its own issues.
Hookworm Na exposure, while safe in controlled settings, can lead to iron deficiency anemia and long-term heavy infections. And the worms themselves can increase gastrointestinal motility, leading to cramps and diarrhea. And frankly, we have to acknowledge the psychological barrier.
The ick factor.
The ick factor. Asking a sick patient to willfully ingest or apply a live parasite is a significant hurdle.
Given the fundamental immune deficit we've identified, are researchers looking at applications outside of just Crohn's and colitis?
Definitely. If this is a systemic failure of immunoregulation, the therapy should work systemically. Celiac disease is a perfect example.
Okay, tell me about that.
Experimental hookworm exposure, Necator americanus, combined with a low-dose challenge of gluten, promoted remarkable mucosal tolerance in patients.
And how did they prove that tolerance? What do they measure?
They took intestinal biopsies and saw a clear shift, decreased expression of the aggressive inflammatory cytokine IFNO, and a concurrent increase in the number of protective tregs. The worm signaled the immune system to tolerate the gluten.
Something the patient's system couldn't do before.
Correct.
And the connection to the massive modern epidemic of metabolic syndrome, obesity, insulin resistance, heart disease, that's also fundamentally an inflammatory state.
It connects directly back to the old friends. Epidemiological studies in areas where helminths are still endemic show that infected individuals have, on average, a lower body mass index, BMI, lower total blood cholesterol, and less aortic atherosclerosis compared to their uninfected neighbors.
So the chronic low-grade inflammation driven by the loss of helminths might be a significant driver in the emergence of these modern metabolic disorders.
It suggests that, yes.
So if the whole worm approach is safe, but hard to prove statistically, and comes with these minor risks and the ick factor, where is the consensus leading the research?
The field is shifting rapidly away from using the whole organism toward identifying and manufacturing the specific molecules, the ICHs, or immunomodulatory components of helminths, that do the therapeutic work.
The goal is the perfect drug, The powerful anti-inflammatory effect without the risks of infection or motility issues caused by the physical presence of the worm.
Exactly. Scientists aim to characterize those specific molecules, the ones that inhibit SMAD7 and induce TREGs and develop synthetic drugs or recombinant proteins to mimic that disease-modulating effect.
Hmm.
They want the tolerance message without needing the messenger.
But you said those molecular messengers are incredibly complex, containing specific post-translational modifications. That sounds like a massive challenge for a drug company trying to reproduce it.
It is the single biggest challenge facing the field right now. Remember, these molecules are native antigens that contain highly specific, evolved structures, those complex glycans and phosphocholine groups.
And you can't just make those in a lab easily.
When researchers try to produce these recombinant proteins in standard laboratory systems like bacteria or yeast, those essential post-translational modifications are often lost because the host organism can't perform them correctly.
So you end up with a bare, simplified protein that doesn't fold correctly or doesn't have the right chemical signature to trick the human immune system.
That's it. The bare, single-defined immunomodulatory products often fail in human trials because they lack the necessary three-dimensional and chemical complexity. They can't replicate the millions of years of chemical coevolution necessary to generate a signal strong enough and complex enough to compel the mammalian immune system into a state of massive, sustainable regulation.
The engineering challenge is replicating nature's complexity perfectly.
That's the challenge.
This has been a remarkable deep dive. We started with the devastating reality of IBD, moved through the historical mistake of losing our old friends, and ended by dissecting the genius molecular hack that these parasites use to ensure their own survival. A hack that could become our medical future.
The central finding is profound. The epidemic of chronic inflammatory disorders, IBD, allergies, autoimmune diseases, and related systemic deficits like reduced stress resilience and metabolic vulnerability in wealthy urban societies is fundamentally an ecological failure.
It's not a personal failure, it's an ecological one.
It stems directly from the loss of microbial and microbial inputs that co-evolved with us, leading to a profound failure of the immune system's regulatory mechanisms.
And the research clearly shows that helminths actively drive the expansion of regulatory T cells by overriding the host's internal brakes, like SMAD7. They restore the natural immune balance, and this molecular insight is moving the focus from the whole organism to the specific components.
And while the use of live worms has proven safe and tolerable, the future is identifying the specific molecules and understanding the microbial shifts they induce. The key challenge remains capturing the complexity of those co-evolutionary signals in a reproducible pharmaceutical format. We have the map, but replicating the ancient language perfectly is the hard part.
So here is a final provocative thought for you to take away. If clean, urban, high-income living inherently reduces the microbial biodiversity required to train a resilient immune system, if we have fundamentally reduced our stress resilience by being too clean, what subtle, everyday exposures, beyond just taking a manufactured pill, must we strategically reintroduce?
That's a huge question.
If the problem is a failure of ecological data transmission to the immune system, the solution might involve changes to urban planning, encouraging specific dietary changes that mimic ancient microbial inputs, or even increasing deliberate exposure to complex natural environments like green spaces and soil. How do we responsibly and safely re-engineer our environment to restore the ancient wisdom that our immune system desperately needs to fight chronic inflammation?
That shift from eradication to ecological restoration is the ultimate long-term goal of this research. It's about learning to live with the biological world again.
A truly fascinating and mind-bending deep dive. Thank you for walking us through that intricate science.
My pleasure.
And thank you for joining us for the deep dive. We'll see you next time.